All right. So I did get a chance to look over the Lab 9 data, and we'll talk about those. And I told you I was kind of up in the air at the Lab Friday about whether I'm going to assign a part two, but I ended up thinking of a idea for part two that I think will work. And so I'm going to talk about that today. So there will be a part two. It's not a particularly in-depth part two, but there's definitely a basic statistical analysis that you'll perform. So I'll talk about the Lab 9 data, and as you can see here on the slides, and if you look at the syllabus, let's look at the syllabus actually. Let's see, here's the syllabus. We are right here. So we're not going to talk about the three-way design stuff at all. If we didn't have the problems with data collection for group project 1, I think I would have talked about lab design today. I would have felt a little bit less pressure regarding group project 2. But we're just going to cut that out. It's not essential for group project 2 anyway, because I think most people will only be doing two-way designs at most complex for group project 2. So the three-way design stuff, just going to not talk about that at all, I think, during the course. We will be talking about Lab 9 and analysis. We're also not going to get to the ERP stuff today. It's okay, though, because we basically have 3 full days of class where that's all we're going to talk about is event-related potential. So we'll get to that. Instead, we're going to finish some of the EEG stuff from last time. So there's a couple complicated studies, old ones from the 50s and 60s that we started talking about with respect to alpha amplitude and its relationship to reaction time. We started talking about those last week. I'm going to go over Lab 9, which concerns alpha amplitude. And then we'll kind of refresh ourselves on these studies related to alpha amplitude and reaction time from last week. And then I'll finish talking about those today. And then that'll be it. And then we'll start ERPs on Wednesday. And then we'll have basically 3 days talking about brain event-related potentials. So still no questions or comments from anyone out there? All right. So let's talk about Lab 9. All right. So for this one, this lab, just to kind of remind you what you did Friday, there was 2 participants. The 2 participants were actually just 2 different versions of me. I did the experiment one day by myself, and then I did it another day with Alex, my assistant. The one with Alex is the one that was recorded. I showed that in class last time. So just 2 different experiments where I was participant for both experiments on 2 different days. And for each experiment, there were 2 conditions. The 2 conditions were my eyes were open, and the other condition where my eyes were closed. And for each participant, for each data file, there were 2 trials for each condition. So there were 4 total trials, 2 for the eyes open condition, 2 for the eyes closed condition. For each participant, for each trial, we calculated alpha amplitude using the BSL analysis 4.1 or the BioPack software. You use the Excel sheet that I provided to calculate the means for each of those conditions for each of the participants. And that's what you copied and pasted into the Google sheet. So I told you when we were doing this Lab Friday that I was going to kind of look at how the data came out. This is the first time I had done this type of a lab, and I just kind of wanted to see how things look. And then I would decide whether we were going to do a part 2 for this lab. And something came up where I decided we should do a part 2, which will concern reliability. So we talked about reliability on a previous lecture earlier in the course. We talked about in particular using calculating Cronbach's alpha, which is probably the most typical measure of reliability. So I used Cronbach's alpha in SPSS to measure reliability. And I did it, there was based on 22 judges. So the 22 judges, let's see how, oops. Right here, judge 1, judge 2, all these judges. There were 22 total judges. When you count all the people where there were scores, there was 22 different judges. So this could either have been a person working by his or herself, or it could have been a group of people that were just submitting one set of measurements. But 22 total judges for this experiment. And close this now. And so I calculated reliability on those 22 judges. And so the term from research methods, this would be inter-rater reliability. This is, or you could say inter-judge reliability. And we're measuring basically the consistency of the judge's measurements. And so one thing I want to point out about these measurements, if you look right here, this line, right, this row right here, this is the participant called EEG alpha demo mat L03. And if you look, you can see this person's data right here. Those are the alpha amplitudes for various judges. And then there's also the participant alpha EEG Rosenthal demo 4 L03, and you can see that person's amplitudes for these judges. Looking right here, it's clear that one of these two participants had overall higher alpha amplitude on that day compared to the other. And that participant is alpha EEG Rosenthal demo 4 L03. 3.5 is bigger than 2.9. 4.79 is bigger than 3.79. 3.500 is bigger than 2.982. And it goes on like this pretty consistently throughout the scores for all 22 judges. And so this is a normal amount of variability that might occur from participant to participant. Some participants, for whatever reason, maybe the attachment of the electrodes, or maybe just the thickness of that person's scalp, or the amount of electricity that person's brain produces. People vary, and the total amount of the strength or the amplitude of the alpha waves that are being measured by the electrodes. And in this case, this participant overall showed more alpha amplitude than the participant right here, even though they were both me. So I would attribute that to the fact that one day I probably had a better connection with the electrodes than the other day. So I got variability in the overall strength of alpha amplitude for these different days, and I made these measurements on myself. And so I'm talking about that because it kind of helps you understand what reliability is measuring, which I know, at least me personally, I don't think I really understood reliability like really well until like the second half of my graduate school, to be honest. But it's also because I didn't get to do activities like this as an undergraduate. And this little bullet right here can kind of help you understand what reliability is measuring for this example. Did the judges consistently measure the smaller score for EEG alpha demo mat L03 and the larger score for the other participants? That's kind of what reliability is, basically quantifying how successful were all of the different judges at making it so that this participant got the low score, and then this participant got the high score. When I measured Cronbach's alpha, our Cronbach's alpha was pretty darn high. I'll remind you, most Cronbach's alpha measurements are between 0 and 1. It actually is possible sometimes for Cronbach's alpha measurements to be negative, although this is pretty rare to my understanding. A very strong reliability is 1. That's basically the strongest reliability you can have, is Cronbach's alpha is 1. So we're pretty darn close to almost perfect reliability, meaning you guys did overall as a class very good at measuring the alpha amplitudes consistently across all the judges. You guys were basically providing the very similar alpha amplitude measurements for these same trials. I will mention I only calculated reliability, Cronbach's alpha, for the eyes open condition. I did not do it for the eyes closed condition, but I'm pretty sure that we'd find a Cronbach's alpha very similar if we would have calculated it for the eyes closed condition. But I want to point out a couple things. I found 2 judges that actually didn't do this. Found 2 judges where they did not, where for them, participant EEG alpha demo mat L03 actually had the bigger alpha amplitude. They basically appeared to, and then for alpha EEG Rosenthal demo 4, these people put the smaller score. So I actually want to show you those people. It's anonymous, so we don't actually know who those people are, but I will show you those. So it's in this one, I believe. Let's go to the end here because that's where they were located. There's one. And there's the red ones right here. So let's look at Judge 31 right here. And you notice the people around him, the top score is always higher than the bottom score. This participant generally had lower alpha amplitude than this participant. When we go to Judge 31, who I highlighted red here, you notice their top score is bigger than their bottom score. Top score is bigger than their bottom score. And once again, notice if the people are just directly adjacent to them, that's not the case. And that's not what I was showing you earlier. So this is the exception. This is one of 2 judges where the top score was higher than the bottom score. And then same thing here, top score is higher than the bottom score. So first off, can someone out there tell me, what do you think was the error? It's pretty clear to me the error that these sets of judges made. Can someone tell me what error was made by these 2 judges? Anyone know out there? Smaller number, higher number. Smaller number, higher number. But then when you get to these judges right here, higher number and smaller number. I have a chat. Let's see what this chat says. They just mixed up the participants. Thank you, Abby. That is definitely what I think happened as well. They were supposed to type the number 3.522 into the bottom, and they were supposed to type 2.98886 into the top. They just reversed the 2. So basically, they wrote this participant on the top row, and they wrote this participant on the bottom row. So yeah, as Abby said, they basically just flipped the participants. We had 2 separate sets of judges that did that. So this would be an example, by the way, of another term from your research methods class, measurement error. When you're making measurements, there are errors that can be committed by both the experimenter and sometimes by technology, though in this case, I actually think it probably were the judges, the experimenters, that made those errors, which can decrease the reliability of the data that you find. And so this is related to lab 2. That's 22 judges, number of items, Cronbach's alpha 0.975. For lab 2, I've made a Google sheet, which is right here. I guess I'll go on and click on it, see how it looks. This Google sheet. And what I've done, when I was originally, the way I copied and pasted, these are what I copied and pasted into SPSS right here, these numbers right here. When I was calculating Cronbach's alpha for the 22 judges, I copied and pasted, these are all of the eyes open conditions for all of the judges. And you can see where I got them from. This cell equals B3. This cell equals D3. And so I'm just taking the stuff B3, D3. These ones that are highlighted yellow, those are all of the eyes open conditions for this participant and the eyes open conditions for this participant right here. And so these are all of the different eyes open conditions for the 2 participants. Please notice, so I've done this for you. I erased these 2 entries right here because they were that first judge, Judge 31, I think it was, that made the mistake where they flipped the numbers. So I'm going to erase those. And basically, and then I'm also going to erase the other judge would have appeared right here. So that was the very last judge. Their 2 numbers were flipped here as well. So they are erased. And you will copy and paste all of these into Excel. And it will look, yes, something like this. You copy and paste those numbers into Excel, and then you will do analyze, scale, reliability analysis, and then you will move over all of those columns where a score appears. The column where a score does not appear, you do not want to move that over into this thing called items. So you're erasing 2 sets of scores. You're erasing the judgments of 2 judges, the scores of 2 judges. And so now when you do the final analysis, there should only be 20 items. And there only needs to be 20 things up here. Each of the columns where there's a score, you'll need to move it up here. But there's going to be that 1 or 2 columns, those 2 columns where a score does not appear. And you'll want to make sure that those don't appear up here. So I'm not going to actually do it because I don't want you to see the answer. I want you to do it yourself. And that will be part of what you do for lab part Lab 9, Part 2. So you're going to have a Word document. And first, I want you to write your prediction as to what will happen to Cronbach's alpha. I basically calculated Cronbach's alpha for the 22 judges. We removed the scores of those two, I'll call them anomalous judges, where the pattern of those scores was not what was the same as the pattern for all of the other judges. And the question I have is, what do you think will happen to Cronbach's alpha? When I calculated Cronbach's alpha for the 22 judges, it was 0.975. I erased the scores of those two judges. And now you are going to calculate Cronbach's alpha. And I just want you to write at the top of your Microsoft Word document, what do you predict will happen? And the reason I'm asking is I just want you to kind of think about how should Cronbach's alpha change when we erase those two anomalous scores? And then I want you to actually calculate Cronbach's alpha. I just showed you how to do it. Calculate Cronbach's alpha and SPSS with those two judges with anomalous data removed. And this is the Google Sheet that I was just at. And then you will paste. So to take I should clarify, I do want you to take a screenshot of, in fact, I'm going to write that here. So I don't want to paste a screenshot of the output into the Word document. I want a screenshot of the output similar to the one that I posted on the previous slide. I basically want to see the new alpha when you are running it with only the 20 judges instead of the 22, when you've removed those two anomalous judges. And I want to see the new Cronbach's alpha, basically the analogous information that I gave you on the previous slide. So any questions about what I'm asking for Lab 9, Part 2? Okay, I don't hear any questions or see anything in the chat. So I haven't posted this on Blackboard just yet, so you can turn it down. I'll do that right after class. All right, so the rest of today, I do want to talk about the means of this real quick, actually. That'll help us segue into the lecture stuff for today on alpha amplitude. So this is the data with all of the judges. So these are the scores for participant EEG alpha dim of this person right here for the eyes open and eyes closed. So these are the means for this participant. These are the means for the eyes open and eyes closed conditions for this participant. We only have a sample size of 2. We have 22 judges, but we only had 2 participants, and I was actually both of the participants. And I calculated using these data on this Google Sheet right here. I calculated the mean amplitude in the eyes open condition by just taking the average of these 2 scores right here. And then the mean amplitude for the eyes closed condition, which is right here. And so overall, what we show is exactly what was predicted based on what I talked about in lecture last week. When your eyes are open, alpha amplitude tends to be smaller. When your eyes are closed, alpha amplitude tends to actually be bigger. I think it's a slightly counterintuitive finding. We think, for instance, when we were awake and your eyes are open, we're engaged with the world, that the brain activity should be bigger or higher or something like that. But it's just the opposite. When we're actively engaged in the world, the alpha amplitude actually tends to be smaller. When we're kind of relaxed and we're not engaged with the world, we're not paying attention to what's going on in there, when we're not taking in information from out in the world, the alpha amplitude actually tends to be bigger. And one of the ideas which is talked about in your some of the papers that you're reading about EEG is that alpha amplitude is kind of thought of as inhibitory, which helps us segue to our lecture, finishing the lecture that we were talking about last time on alpha amplitude and its relationship to reaction time. So the idea is that let's see, where did we leave off last time? I think it was this slide right here. Where is that slide right there? Yep, that's where we left off last time. So those are the data we were looking at the end. I'll kind of go back just so we can kind of remember. I just showed you that in our lab, when the eyes were open, alpha amplitude was smaller. When the eyes were closed, alpha amplitude was bigger, which is basically this. Alpha blocking is, or alpha event related to synchronization is the idea that alpha amplitude decreases. That's about event related to synchronization, or event related, or alpha blocking is just a decrease in alpha amplitude. That's what happened for us when we opened our eyes, or for me at least, when I opened my eyes and was starting to be attentive to the world around me. That caused alpha blocking, or event related to synchronization. There's relatively less alpha blocking when your eyes are closed, because that's when alpha is biggest. So this is the article I was talking about last time. It was kind of looking at some of the behavioral, if there are behavioral correlations with the amplitude of the alpha amplitude. Basically, the idea is, when you, based on what we just talked about, the eyes open, eyes closed experiment, basically it appears that when alpha amplitude is really big, this means that you're not very attentive to what's going on in the outside world, which means that when alpha amplitude is big, if you're not attentive to what's going on in the outside world, then your ability to respond to something out in the world should be slower when your alpha amplitude is big. Conversely, we saw that alpha amplitude was small when our eyes were open, my eyes were open when I was engaged with the world. And basically the prediction would be that when a person's alpha amplitude is small, this would predict that their ability to respond to something out in the world would be quicker. They'd have smaller reaction times when the alpha amplitude in their brain is small. So this is the article I was talking about last time. I feel like we need to review it, because the paradigm, the variable 4-period paradigm, it's kind of old school. There are actually some studies in the 21st, several actually in the 21st century that have been published with this, but it is kind of an old school, nonetheless, paradigm, the variable 4-period paradigm. So I doubt most of you have heard it in other classes. And so I'm going to review it, even though I did talk about it last week. I just want to review it because it makes some kind of counterintuitive predictions. And as I said, you probably haven't heard much about it. So in a variable 4-period task, there's a warning signal that occurs, and it just tells you in the near future a stimulus will be occurring. And then in the near future, indeed, a stimulus does occur. And your job is, when you hear that target stimulus, just respond by pressing some button as quickly as possible. The dependent variable is reaction time. In the variable 4-period paradigm, the duration of the 4-period varies somewhat randomly from trial to trial. So sometimes there would be the warning signal, and then there would be only a short duration before that target signal occurs. And then sometimes there'd be a warning signal, but there'd be a relatively longer duration until that target signal occurs. And the question is, what happens to alpha amplitude during these two different conditions, when there's a short duration between the warning signal and the target versus a long duration? And what also happens to reaction times? So the general results for variable 4-period effects regarding reaction times, the behavioral measure, is that the longer the duration of the 4-period, the quicker the reaction times will be. And so this is a thing that conceptually I kind of want you to understand the logic, why the researchers, this is a very common hypothesis in variable 4-period research works, why the researchers thought that this was a reasonable hypothesis. And the idea was that over the course of the experiment, the participant starts to learn, okay, sometimes there's a short duration before the target stimulus, and sometimes there's a relatively longer duration. On any given trial, the participants kind of like, okay, will this be a short duration or a long duration trial? Will I have a little time to wait before the target stimulus, or a long time? And if there's a long 4-period duration, think about the participant kind of waiting. There's a warning signal, and then a short duration passes, and the participant goes, okay, the short duration has already passed, so I know that the target stimulus is not going to occur during the short duration, which means, oh, it must be occurring in that longer duration that I heard. So the duration between the warning signal and the target, it's going to be the long one this trial. And I know that because the short duration already passed. And so that long one has to be the right answer. It's going to be a long trial because I already know from the process of exclusion. It can't be the short one because that short duration of time has already passed. Therefore, the correct answer has to be the long duration. And so in theory, this gives the participant high amounts of certainty regarding when exactly the target's going to occur. You never get that for the short durations because as soon as that warning signal occurs, there's two possible futures. The 4-period duration can be short, or the 4-period duration can be relatively longer. But as soon as that short duration passes and the participant knows, okay, the target signal is not going to occur after a short 4-period duration, the participant knows it has to be a long 4-period duration, which gives the participant more certainty, which allows the participant to prepare their mind and their body better for the long duration 4-period compared to the short, which should result in quicker reaction times for longer duration 4 periods. So that's the typical theoretical logic for this hypothesis. Longer duration 4 periods result in quicker reaction times. And if we think about what we've been talking about with alpha amplitude already, that should mean that for these longer duration 4 periods, alpha amplitude should be smaller because we already saw alpha amplitude gets smaller when we're more engaged with the world. When we're more engaged with the world, we should respond to it more quickly. So this makes the prediction. Alpha amplitude will be smaller on those longer duration 4 periods conditions, and the reaction time should be quicker for those conditions as well. So I showed you this last time as well. This is the warning signal. This is the stimulus. And then this is the participant, the participant's response. So I think I have a question here. Let's take a look. Yeah, that's a great question, Ahmed. So Ahmed asked, what if the duration is really long, let's say a few hours? So I don't know what would happen with a few hours, but I'm actually going to show you a study that was published later on, like 10 years after this one, where that's exactly what they did. They didn't extend the duration by a few hours. They just extended it by a few more seconds than usual, and they did find a completely different pattern of results. So that's a really good question. Your intuition's good. The total length of duration of the 4 period does matter, and it changes the results in important ways. And I'll show you that in a second. Okay, so here's a long 4-period duration because this duration is long, which results in a relatively small response time. You can see that this amount of distance between the target stimulus and the response is small here as the 4-period duration is long. Here, the 4-period duration is quite small. There's the target stimulus, and we have a relatively longer response time here than we did up here, where the gap is smaller here, relatively longer here. So this is consistent with what I told you. This is the behavioral study, this is the paradigm for the Lansing study. And then I'll show you the EEG and behavioral results on the next slide. These are basically the predictions, what they predicted to happen. So the important stuff is down here, the difference between the two 4-period conditions. So they had one condition where the 4-period was short, between 50 and 250 milliseconds. So that's the short duration 4-period. And then they had the relatively long duration 4-period, which was right here, where the target, the amount of time between the warning signal and the target would be longer, 300 to 1,000 milliseconds, versus the short durations, 50 to 250 milliseconds. So the study looked at alpha amplitude and reaction times. And first, I'll talk about the alpha amplitude. I did talk about this briefly last time, but I'm going to go to a little bit more detail than I did when I talked about it Wednesday. So here's the short 4-period duration, short variable 4-period duration. And they said the alpha was not blocked. You got to think about why that kind of makes sense. Because, as I mentioned, this is the situation in the short variable the short 4-period duration, where the participant never gets to kind of adequately prepare for the possibility that the thing might occur very quickly, where they have less time to prepare. And so alpha is not blocked. Alpha was big, in other words. Alpha amplitude was big because alpha was not blocked. It was still big. And what that indicates is that the participant was not in a high preparatory, or what the interpretation of this was, the participant was not in a high preparatory state. Their mind was not adequately prepared for a stimulus in the short 4-period duration, at least based on alpha. But if you look at the reaction time, they see a relatively big reaction time for that condition as well. For the longer 4-period duration, they said the alpha was blocked, which basically means, this is kind of an old school way of presenting data, but basically what this means is that alpha amplitude was significantly lower in this condition, the long duration, than it was in the short 4-period duration. They showed a significant difference in alpha amplitude between these two conditions. It was smaller here, and it was relatively larger here. Behaviorally, they also showed that in this condition, the reaction times were smaller. So for the long 4-period duration, alpha amplitude was blocked, it was small, and reaction times were quick, they were small as well, where the opposite was true for the short 4-period duration. Alpha amplitude was relatively bigger, and reaction times were also relatively bigger as well. So this study, which is now 60 years old, was originally interpreted as saying, yeah, alpha amplitude seems to be really important for preparation, and more, and engagement with the outside world. When alpha amplitude is small, that means your mind, your body, your brain are prepared to respond to something out in the world, which should cause you to react to it quickly. But then there was a study that was published. When was this other one published? This is Levitt, 1968. So about 9 years later, another study was published that basically called out the original Lansing study, 1959. And they said, we have data that suggests that this original interpretation regarding alpha amplitude is problematic. The alpha amplitude might not actually reflect a participant's degree of preparedness, the amount of engagement that they have with the in the outside world. Theoretically, part of the reason that their their kind of complaint makes sense is because of other findings that have occurred, related since this study was presented, such as this right here. It's actually now known, this has been way studied since then, and this is a very highly replicable effect, that when you present a warning signal that's auditory to a participant, it does kind of several things, but those several things take a while before they go into effect. The main thing that happens, and we know this occurs in the brain now, is that when a sound occurs, there's a lot of activity in motor regions of the brain. So those same regions of the brain that become activated when you're moving your hand, or when you're speaking, or moving some body part, those same regions of the brain get activated when you hear sounds, which is interesting because other sensory modalities, like when you see something appear, that doesn't necessarily cause your motor system to become activated. But when you hear sounds, there's something about sounds where they kind of prime your motor system. They basically engage your motor system. And this has been shown that when you use auditory warning signals, it basically can make it so the body responds more quickly to things in general. But there's a couple things which are important about, compared to a visual warning signal. When you have a visual warning signal, it doesn't prime the motor system, and so you don't get these as much speed increases. But for just an auditory warning signal, it primes the motor system, the motor regions of the brain activate. And there's one thing which is important about that is these effects take about 300 milliseconds before they go into effect. You hear a sound, and then a certain amount of milliseconds later, the motor regions of the brain become activated. But it takes a few seconds, or a few milliseconds before it starts. And also, just as importantly, is it's transient. The motor regions of the brain become activated when you hear a sound, but it lasts for just a short period of time. So that phenomenon actually is probably, I'm kind of giving you the conclusion of all of this before I actually present you the study of Levitt, 1968. That phenomenon probably could explain all of the findings from Lansing et al. 1959. So this is kind of, in other words, the correlation between reaction time and alpha amplitude, or event-related desynchronization, it might have just been a coincidence. And it's a coincidence that was driven by this idea that sounds kind of temporarily stimulate the motor system. And that's why there was the increase in reaction time with the long 4-period duration only, because it takes a few milliseconds before that priming of the motor system occurs. In this study, the Levitt study, they wanted to know what happens if we have really long 4-period durations. And one thing that is important is that motor priming that occurs when you hear a sound, it's transient. It only lasts like a second or two, which means that benefit of reaction times that you get might actually go away the longer that you have to wait. So if you have a variable 4-period and you're waiting for longer than just a second or two, really you might lose that kind of quick reaction times, because in reality, the thing that was causing those quick reaction times might have been something else, such as priming of the motor system by sounds. And that priming is transient and only lasts for a second or two. So yeah, this is kind of what Levitt did. They performed similar experiment, but they extended the 4-period duration here just to 4 seconds. Zach might ask that good question earlier, several hours. I don't know what would happen if you extend the 4-period duration several hours, but I do know what will happen if you extend it several seconds. So just to remind you, for the original study, the 4-period duration, the longest one was 1 second, 1,000 milliseconds. For this study, Levitt, 1968, they extended the longest 4-period duration to be 4,000 milliseconds, which is 4 seconds. And so let's look at what happened. Once again, these are old school studies, and so they just presented data they just were not as efficient or not as good, not as clear in the way they presented data back in the day, that you can still kind of work what was going on out. It just takes a little bit more scratching of the head to kind of understand the figures, but I'll kind of help guide us through this. So first thing, they did kind of replicate the effect to an extent. They replicated the original effect from Lansing et al. And if you look at Lansing et al., they had a super short 4-period duration in Lansing et al., and they also had a relatively longer 4-period duration. So 200 is very short, 500 is relatively longer. And if you look at what happens to reaction time and alpha amplitude for these two, this totally is the exact same thing. This replicates quite nicely just for these two conditions, the short and the medium to long, what happens to alpha amplitude and reaction time. So let's look at each of those. So let's look at reaction time first. Reaction time for the very, very short 4-period duration for this study, Levitt, 1968, was relatively higher than it was for this 4-period duration right here. So in other words, they're showing the kind of the typical variable 4-period effect. And that's reaction times for the 500 millisecond 4-period were quicker than for the 200 millisecond 4-period. That's the same thing they showed behaviorally in this Lansing et al. Study right here. And they also showed an analogous thing, basically the same thing regarding alpha amplitude. So they used event-related desynchronization, which adds that extra level of it's a little bit harder when they use ERD as kind of the dependent measure. I like it better when they just say alpha amplitude, because alpha ERD, remember, that's basically saying the dependent variable is a decrease in alpha amplitude, which is cognitively a little bit harder to kind of think about. But that's what they're showing here, basically, the percentage of stimuli that occurred when the participants' alpha oscillations were really small. That is, when they were having event-related desynchronization of the alpha band. And so what's confusing about that is here in the 500 millisecond condition, the alpha desynchronization is really big. But when alpha event-related desynchronization is really big, what that actually means is the alpha amplitude is really small. So it's a confusing figure. That's the kind of just the way things were back in the day, unfortunately. Come a long way since then, I would say. So once again, I feel like I just need to repeat this because it's so complex. Event-related desynchronization was small here, which means that the alpha amplitude was big for the short 4-period duration. For the relatively longer 4-period duration, the event-related desynchronization increased, which means that the alpha amplitude got smaller. And so for these first two 4-period durations, exact replication of what they found. Basically, the longer the 4-period, the smaller the alpha amplitude. The longer the 4-period, the smaller the reaction times. So these two conditions by themselves kind of support the same old thing that Lansing et al. Proposed, which is when alpha amplitude is small, that means your mind is more prepared to respond to something which is out in the world, and you respond more quickly to it. But then things get a little bit wonky after that. And that's because of what happens right here, where for these longer, really long 4-period durations, so 1.5 seconds and 4 seconds, and then we lose, right, and it says right here, they didn't replicate the typical variable 4-period effect when considering the 1500 and 4000 millisecond 4-period durations. So what you would have expected based on the theory of variable 4 periods is that alpha amplitude should continue getting, so here they're showing alpha event related desynchronization. So what they would have predicted is that this line, the dotted line, keeps getting higher and higher and higher the longer the amounts, the longer the duration of the 4-period is. Because in theory, the longer the duration of the 4-period is, the better prepared you should eventually be when that target comes. And if that preparation is just getting better and better and better, the event related desynchronization should be getting higher and higher and higher, which is another way of saying the longer the 4-period duration, what you would have predicted is the alpha amplitude would continue getting smaller, smaller, and smaller, and smaller, and smaller, depending on 4-period duration. That is not what they found at all. They also would have made the prediction regarding reaction times, which is easier to understand, I would say. They made the prediction that the longer the 4-period, the shorter your reaction time should be, because the better prepared you'll be for those really long 4-period durations. But that's definitely, they didn't show really either of those. So right here, things are these are the reaction times. Right here, once again, I showed you this earlier, the reaction times got quicker between the very short and the medium 4-period duration. But then as the 4-period durations increased, the reaction times went back up. And then also the alpha amplitude, the desynchronization was decreasing, which basically means that what this is saying, they had a quick, for between the 200 to 500 milliseconds, a quick increase in event-related desynchronization, which basically means there's a quick transient decrease in alpha amplitude. But it was transient. And then by about a second to 1.5 seconds later, 1.5 seconds later, alpha amplitude was basically increasing again. So alpha amplitude decreased right here, alpha amplitude decreased right here, and then it started increasing for the remainder of the duration of the experiment. And so this really supports the kind of the theory which has emerged since all of this research, that really one of the main things driving these variable 4-period effects for the really short 4 periods is kind of just like a transient priming of the motor system. It takes greater than 200 milliseconds after the warning signal before the motor system is primed, which is what occurs right around somewhere after 200 milliseconds, but before 1500 milliseconds, the motor system is temporarily activated just by hearing a sound. But then that activation goes away, and then your motor system is no longer primed, which results in the reaction times going back up and the alpha amplitude going back up as well. And so this really kind of throws a wrench in the original findings, because these variable 4-period effects, basically, they're just really short. But then for later variable 4-period durations, the effect goes away basically completely. And the relationship between the alpha amplitude and the reaction times gets much more complicated as well. So here is kind of the final results of this Levitt study right here. And they basically showed no significant effects. So once again, they present these data in these old studies in kind of ways that are a little bit weird. They're also using, I'm pretty sure that this study was actually published before ANOVA had even been discovered yet. So F, which we all now do all the time, F, I'm pretty sure had not even been discovered, or if it had, it was like a hot new thing at the time. And so they weren't even using F, and they weren't using T for that matter either to compare the different conditions. So the statistical analysis here are way different than what would be considered acceptable nowadays. But let me talk a little bit about what they did. So their dependent variable, once again, was a little bit weird. The median percentage of trials introduced during evoked alpha desynchronization. This basically means what percentage of the trials was the alpha amplitude really small compared to what percentage, that's what these numbers are right here. The percentage of trials which occurred when the alpha amplitude was really small. when that target stimulus was presented. Was the alpha amplitude small? That's what these numbers are, basically. The more trials where the alpha amplitude was small when that target signal was presented, the higher the number will be right here. And then they compare the amount, they're basically comparing the amplitude of alpha between people that were fast, their reaction times were very quick, and the people whose reaction times were very slow. And you would make the prediction that there should be more people in the fast responders group that would have trials with alpha desynchronization, because once again, alpha desynchronization means your alpha amplitude is small. And you would expect that the people that were really fast would have smaller alpha amplitude on average than people who were slow, but they showed no significant difference here. No significant difference. There was a trend right here for the fast responders for them to be slightly higher. Fast responders and slow responders. So there's slightly more people who were fast that had smaller alpha amplitude compared to the slow responders, but that difference was not significant. And then when we look at the really long 4-period duration, it's kind of like the opposite effect for really long 4-period duration. People that were really slow responders actually had more event-related desynchronization. That means these people were slow, yet their alpha amplitude was actually really small. And that actually was that was marginally significant. So I guess Z is greater than 2.33. P is not. I think that means that's not significant, I think. But it's close, but it doesn't matter because it's not the predicted direction at all right here. So they did not support overall, did not provide any support in this study that alpha amplitude predicts reaction times. So there's kind of this controversy regarding the interpretation of the results, and it's still a controversy that's kind of lingering today, which is why we're reading some of the more recent articles. I just presented you these. I was talking about the old EEG articles last week, but the articles that we're reading about EEG, like the EEG Review 1, and then we also have EEG 1 and EEG 2, which are related to this review article. This is some of the stuff that's kind of looking at how the literature has evolved in the years since those original studies. Not as much work on the variable 4-period effect anymore, but people are still interested in what does alpha band amplitude, what does that have to do? How does that relate to our behavior? You know, performing different types of mental tasks, like paying attention to different things or having to remember things in short-term memory, which I've talked about these studies and you've read about them and you've taken a quiz about them. What is the relationship between alpha amplitude and performance in these different types of tasks? So I think that's where we're going to leave it off. I actually have already talked about this last time and you've read about these. So I think I'll just let you have the rest of the time to work on Lab 9, Part 2. And I do believe I have a meeting with one of the groups and I'm happy to meet with those people now. But I'm going to stop recording now.